Today lecture, guys. On Wednesday's lecture, we started talking about gene regulation at the level of transcription. And we went through some of the very key concepts. And we said that to understand that gene regulation is mediated by transcription factors binding to specific sequences and in combination with other transcription factors, determine the amount of transcription from a gene. So to understand gene regulation in an organism, first we need to find out what are the transcription factors in that organism and which sequences do these transcription factors bind to, right? And in the last lecture, we went through some of the techniques that molecular biologists use to look at which sequences do specific transcription factors bind to, such as the footprinting assay or gel shift assay and other techniques like that. And one of the techniques that we talked about was the reporter assay, which is going to come again and again in throughout these lectures because you can use reporters to look at a whole bunch of different stuff. Okay? So that was the first part. And now we are going to learn how the transcription factors themselves are regulated. All right. So first we're going to talk about the fact that transcription factors are modular in structure. That means there are distinct protein domains that do distinct functions. And how do we know this, right, first of all? All right, first let me tell you what are the two typical domains that we see in individual transcription factors. Here are some transcription factors from yeast. We already talked about GAL4 in the context of GAL4 recruiting the SALGA and other chromatin remodeling complexes to genes to activate transcription. And what we said was that usually, not usually, always, sequence-specific transcription factors that bind to DNA contain a DNA binding domain that is separable from another domain that is used for activating transcription. So that domain is called the activation domain. And these two domains are usually linked by a flexible region that connects them in the protein. All right, so, and also that transcription factors can have more than one activation domains. All right. So how do we know this? We know this through some of the earlier reporter assays that looked at the ability of different protein regions to do their function. So the necessity and sufficiency of a protein domain to be able to bind to DNA or not. So how do you do this, right? So the first thing that you do is you take a reporter gene, you construct a reporter gene that you know that your transcription factor regulates. Let's say we want to know the protein DNA binding domain and activation domain of GAL4. We know that GAL4 in the yeast binds to a DNA sequence called the upstream activator sequence called GAL. And what we can do is we can take that upstream activating sequence and clone it right upstream of a basal promoter. That is a promoter that's going to support a little bit transcription, but not a lot by itself. Only if the transcription factor GAL4 binds to the UAS, then you're going to have a high level of transcription. And then what you do is you can clone that in front of a reporter gene. And in this case, the reporter is LAC-Z. Okay? And then in the same cell, what you're going to do is you're going to express different versions of the GAL4 protein. So as a positive control, that is in a control where you should see the actual binding and the activity. Activity means transcription activation. This full-length protein should be able to bind to the UAS and activate transcription, right? So if you transfect your cells with this reporter construct, which is here, along with a construct that is going to be used to express this full-length protein, you're going to see the transcription activation And you can also assay if that full-length protein is able to bind to DNA using one of these assays that we talked about in the previous lecture. And then what you can do is you can put a plus if it binds to DNA, and then you can put varying numbers of pluses for activation of transcription. Okay? And in the positive control case, the full-length protein is going to bind and activate. All right. So what do we want to do next? We do a very similar idea of how we actually learned what part of sequence is important, etc. Here we're saying what part of the protein is important. So the first, the strategy that you would use is you make truncated Gal4 protein. Okay? So you make small deletions in the protein and see what the effect of that deletion is. So let's start with the first one here. Here is the full length, so that's our control. It is able to bind to DNA and it's able to activate transcription. And now what you can do is you can make a small deletion starting from the N-terminal of the protein and then say, is this truncated protein here capable of binding to DNA and activating transcription? And what you can see is as soon as you went in and deleted 50 amino acids from the N-terminus of the protein, that you found that there is no binding at all. And a transcription factor cannot activate a specific gene if it doesn't bind, right? So if you delete the DNA binding domain, of course, you're not going to get any activation from the reporter. Okay, so then what you can do, you can make serial deletions to narrow down the regions that are important for each individual activity. So you decide, okay, the left end of the protein and terminal end of the protein is DNA binding. Let me find the activation domain, and let's start from the C-terminal, right? Because we know N-terminal is probably going to bind the DNA, and cut the protein into smaller and smaller pieces. So you make a truncated protein that's a little bit smaller, and you find that did not affect the binding or the activation. You make a little bit more deletion. It still did not make any difference. So you know, okay, these regions of the protein is not important for the activation. And you make a little bit more deletion, and now you started affecting the activation because you see a little bit less transcription of the reporter, and you make more and more deletions, and you can see that now the activating ability of the protein is gone. But it is still capable of binding to DNA because it still contains the DNA binding domain at the N-terminus, right? So by looking at these coordinates, what you can see is the first time that you started affecting the transcription, you started deleting something between 792 and 823, right? So what you can say is starting from here is the activation domain starts, and then you can, but you can't say what on this side, what is the coordinate on this side where the activation domain stops, right? And to do that experiment, then you need to do deletions from the middle of the protein. So you're going to make truncated protein, but you're going to delete the in the middle. So what you can do is you can start deleting the protein from starting from the middle, and now you can make further and further deletions inside, and you can see that when you start deleting sequences, deleting amino acids that are after 738, you started affecting activation. So you know that anything between 738 and 823 is where the activation domain is. Okay, so you can also make deletions this way, right? And here the results of that experiment is not shown, just the end result is shown. And the end result shows you that anything from the N-terminal to the 74th amino acid contains the DNA binding domain. And the reason I can say that is that if you only take that, right? If you only take that 74 amino acids, and if you take the region of the protein containing the activation domain, you can bind to DNA and you can activate transcription just as well as the wild type. So you know that this domain is sufficient to bind to DNA, and this domain is sufficient to activate transcription, right? Necessity and sufficiency. These are different things. Does that make sense? Question about this reported assay. And what's important is that these two activities are separable. So you can make truncated proteins that can bind to DNA but not activate transcription, right? But you can't make a protein that doesn't bind DNA and not activate transcription. But we know that that's also possible if you took the DNA binding domain of another transcription factor and swapped in with the GAL4 activation domain and targeted to a reporter gene, then it's going to be able to activate transcription. Does that make sense? So if you artificially target it, the activation domain can work independent of the DNA binding domain. So these are two separable activities of a transcription factor. All right. So that's how we know that transcription factors are modular in structure. And so the transcription factors in humans and animals contain DNA binding domains that are made up of certain specific structures that evolve with time with having diverse amino acid sequences. But they fall into different categories based on the DNA binding domain structure that recognize the DNA and binds to it in certain specific ways. So here are some examples. So this is the family of transcription factors containing DNA binding domain with basic loop helix loop helix domain. And what you can see is that there is a helix here, and there's a loop. There's a helix here, and then there's a loop, and then there's a helix here, and then there's a DNA binding helix here. And this transcription factor DNA binding domain binds as a homodimer. You can see that there are two of them. And you can see that the helices that are recognizing specific sequences in the DNA are sitting at the major groove. You can have other DNA binding domain structures where you have a helix-turn helix structure recognizing the sequences. These are interesting. This is an interesting family of transcription factors. They have zinc fingers, and these zinc fingers can sit in the DNA and recognize a few sequences at a time. We're going to talk about this in genome editing lecture a little bit more, and how you can utilize this kind of structure in certain applications. And there are other transcription factors such as leucine zipper factors, where you have a certain structure where you can the two dimers zip by interacting, and then recognize the DNA with the loops. All right, so these are the type of transcription factors, some of the families of transcription factors. You may have heard of a transcription factor family that is pretty famous, because when they're mutated, they give these wild phenotypes, which are awesome, like legs coming out of your eyes, right? And that's like the classic picture of a fly that you would see in a genetics class or your POB, because it just is amazing, right? Not only the fly doesn't have eyes, it has legs coming out of its eyes. How is that possible? So the transcription factors that control that development of limbs belong to a family called the homeodomain containing transcription factors. So the homeodomain is basically a DNA binding domain that sits into the DNA major groove and recognizes somewhat this consensus sequence, TAA, TTA, right? So the homeodomain transcription factors contain a homeobox. Don't confuse this as a DNA sequence where things bind. This homeobox is the region of the coding region of the protein that encodes for the homeodomain, okay? So then you might hear in the future homeobox containing transcription factors. You'll immediately say, okay, these are the homeodomain transcription factors. So, and they're involved in the regulation of anatomical development, morphogenesis in animals and plants and fungi, etc. And what's amazing is that the same type of transcription factors that control the development of limbs in flies are the same type of transcription factors that control our own development. So these transcription factors evolved with time for somewhat different DNA sequences, binding to different sequences, but their function has been conserved, right? And that's why we study flies. All right, so this is the guy I'm talking about, and here is the Antennapedia mutant, where you have, not the legs are not coming from the eyes, they're coming out of the antenna. So instead of the antenna, these guys have legs. All right. They still have eyes. Okay, so the transcription factors are categorized into different classes based on their DNA binding domain and the DNA binding domain structure. So to understand transcription, next what we want to know is which you basically have isolate. Let's say that you've done a reporter assay in which you determined a region of the upstream sequences that you know is bound by a transcription factor and regulates gene expression, right? When you isolate that sequence, you actually don't know what transcription factor binds to that sequence, right? You know that it's important, but you don't know what binds to it. And how can you actually find that out? So ask the question, which transcription factors bind to my regulatory sequences? And one of the classical techniques to do this is called the yeast-1 hybrid assay. This is a mixture of, it could be a candidate transcription factor or it could be a transcription factor library. Okay. So how do you do this? What you do, the principle of this is here. What you first do is you take your DNA sequences that you know that some transcription factor binds and regulates transcription, and you clone it upstream of a reporter gene. Okay? So just like in the previous reporter assay, if your protein binds to this site, whichever protein this might be, we don't know, what's going to happen is it's going to activate the transcription. Let's say that you saw that in your regulatory sequences, you saw a bunch of TAAT sites, right? And you said, this could be a homeobox containing transcription factor. So what you can do is you can go to your genome and say there are 20 of them I know. So I'm going to do a candidate assay and see which one of these actually bind to it. So what you can do is you can actually clone your gene, candidate gene, in a plasmid that is going to support the expression of this gene, and when this gene is translated, it's going to make that transcription factor of your, the candidate transcription factor. Now, if you transfect your cells with both of these plasmids, they're going to go into the nucleus and make your protein. And if your protein binds to the regulatory sequences in your reporter plasmid, then you're going to see the expression of the reporter gene, right? Does this make sense so far? Any questions? All right. So what next? What you can do next is, if you don't have some candidates or your candidates were not the right transcription factors to support the transcription, you can actually screen a library of transcription factors. How do you do that? First, you need to construct a library. And when we construct the library, we can take advantage of the fact that the transcription factor DNA binding domains are separable from the other parts of the protein. Okay? So what you can do is you say, in humans, there are 2,000 transcription factors, more or less. you say, here are the DNA binding domains, and you can identify those domains because you know a lot of transcription factors belong to certain families, right? And those DNA binding domains have some kind of conservation. So by doing a sequence comparison, you can say, that transcription factor, this region looks like a helix-turn helix, so that's probably where the DNA binding domain is. And you say, I'm going to clone that region of the coding region of that and into a plasmid. So here, in yeast-1 hybrid assay, what you can do is you can construct plasmids that are going to express your transcription factor or the DNA binding domain of your transcription factor that is fused, the protein is fused to a yeast activation domain. Okay? So when these plasmids are going to express your transcription factor, they're going to express your full transcription factor or the DNA binding domain that you cloned that is fused to a yeast activator domain. Does that make sense to everyone? When I say fuse, do you understand that it becomes a single protein? And one part of the protein is what you cloned, that is the DNA binding domain, and the other part of the protein is the activation domain of a yeast transcription factor. Does that make sense? Okay. That's important that you understand that part, okay? Then what you can do is you can construct a library of plasmids that contain every transcription factor that you want to assay. So this could be all 2,000 transcription factors from humans. And now what you do is you also make a reporter plasmid in yeast. So in yeast, you can use the yeast auxotroph mutants. These are mutants, unless you provide a certain amino acids, cannot live in the plate, right? So I think you learned this with Professor Siegel. Is that right? Did you guys learn it? Because, yes, right? Okay, thank you. So what will happen is that if this his3 gene is expressed, and you take these yeast cells and plate on plates that do not contain any histidine, these cells will be able to grow, right? And if it's not expressed, because there's no histidine in the media, these cells are not going to grow. So you can monitor the expression of the reporter very easily by plating these guys on plates. And now what you're going to do is you're going to put your sequence that you want to know which transcription factor binds. And now in this assay, we call this sequence bait, okay? Because we're going to take that bait and go to our pool of transcription factors and fish out which one actually binds to it, right? All right. So that's our bait. And now what do you do next? You do take each of these library plasmids that are in your transcription factor library, and you're going to transform them, the yeast, with the reporter plasmid. And if your DNA binding domain or transcription factor binds to your bait sequence, because it is now fused to the activation domain of a yeast transcription factor, it's going to be capable of activating transcription. So these yeast cells are going to can live on histidine plates, right? So if you do this with a whole bunch of all of the plasmids in the library, and then isolate the yeast cells that have lived, and go back and isolate the plasmids, then sequence the plasmid, then you know which of the transcription factors that you cloned is binding to the target sequence, bait sequence. All right, does that make sense? All right, so this is how we find the candidate transcription factors. So now let's talk about how are transcription factor binding and function regulated, okay? And there are several factors here that we're going to talk about. We're going to talk about how chromatin structure regulates transcription factor binding. We're going to talk about the fact that transcription factors can work in combination with each other. And we're going to talk about that some transcription factors being regulated by ligands, just like how bacterial transcription factor is regulated. Give me an example. in which ligands regulate transcription factor binding in bacteria? We just did prokaryotic transcription and had a midterm. LAC repressor, TRP repressor. These are transcription factors that are regulated by ligands, right? So one thing that I recognize with you guys is you might see that I always ask questions about other lectures. The reason why I do that is the lectures are not just individual meals. These are not just dinner, right? The ability to understand the lecture and connect between them is what is going to make you really strong in molecular biology. Okay? So try to think widely the whole semester, not just the topic that we're talking about. All right. So how does chromatin structure regulate transcription factor binding? So I showed you some crystal structures of the DNA binding domains, and what I showed you is that there are actually alpha helices that are sitting right onto the minor groove and something else grabbing the DNA on the other side. So it's really making strong interactions with the DNA. But those interactions, for most transcription factors, not all, are disrupted and cannot be formed if the DNA is wrapped around the nucleosome, right? And here's the nucleosome here, where you have the histone octamer sitting in the middle, and the 150 base pair DNA is wrapped around it. And here's the side view. And imagine now you have a transcription factor containing multiple zinc fingers that need to sit on the DNA on this end, on these sides. It's not going to be able to do that when the DNA is like this, right? So nucleosomes, the nucleosome position and occupancy at a site is then going to affect the ability of a transcription factor to bind at that site. So how are the nucleosome positions and occupancy in the whole genome? So a lot of whole genome chromatin mapping assays, which I'm not going to go through, but one of which is CHIP, chromatin immunoprecipitation followed by sequencing. We talked about it in last lecture. And here's a question. See if you guys can connect this. If you wanted to use CHIP to figure out where nucleosomes are located in the genome, what would you do? How can you do that? How can you use the CHIP-seq technique to map where the nucleosomes are? What's the first step of the CHIP-seq technique? You take your chromatin, you cross-link it, you sonicate into individual pieces, and then you need to immunoprecipitate, right? This is called chromatin immunoprecipitation. Give me, what antibody would you use at that step to map nucleosomes? Excellent. You would want, if you're mapping nucleosomes, you will want an antibody that specifically binds to one of the histones, right? Because that's what protein you're going to map in the genome. And experiments like this have established that in eukaryotic genomes, the nucleosomes are not randomly distributed in the genome. What they have found is that a lot of the coding regions and introns and exons contain a lot of nucleosomes that are positioned somewhat regularly, but the promoters contain less stable nucleosomes. So these nucleosomes can come on and off from the promoters, leaving them more or less open compared to the coding regions. Okay? And so what happens at the promoters is that sometimes transcription factors can find a window of opportunity to bind. So what that does is that transcription factor binding sites that happen to be at the promoters will be utilized more. They're going to be bound more compared to transcription factor binding sites that might be inside the gene. Okay? So that's how nucleosome occupancy can regulate transcription finding in the gene, transcription factor binding in the genome. All right. I just told you how transcription factors might find the promoters, and you might ask, what happens at the enhancers? we know that transcription factor binding to enhancers in eukaryotic transcription is very important to control the transcription of that gene. So if there is a nucleosome sitting at the enhancer, then how do these transcription factors get to this site? Okay? So this is a problem that's not solved, but so far what we know is that, for example, in very early embryogenesis in flies, the genome, the enhancers that need to be used to start transcription of some of the critical transcription factors are closed, okay? But soon after the zygotic transcription starts, zygotic transcription means transcription of the zygotic genome, right? There is a protein called ZALDA that is a pioneer transcription factor. So a pioneer transcription factor are transcription factors that can bind to their DNA sequences even when the DNA is wrapped around the nucleosome. So what this ZALDA protein does, just like many transcription factors, is that it binds to the nucleosomal DNA and somehow it opens up the enhancer, so its binding results in lack of nucleosome at that site. Now that when it does that, it allows other transcription factors, such as transcription factor bind, to now come and bind to this enhancer. Okay? So in this case, in this example, TF2 is a pioneer factor that clears out the nucleosomes so that the other transcription factors can bind. This is a very critical concept, not only for fly development, but also human development. A lot of the cells that are going into different fates to become to make the liver, to make our muscles, to make our brain, etc., contain a set of pioneer factors that initiate these processes. Okay? All right. How can a pioneer factor open up chromatin? There are a couple of concepts here. One, these are still working hypotheses in the field. One of the hypotheses is this. The nucleosome, to be able to bind to DNA, it's going to bind to about 150 base pair stretch of DNA. Okay? But if you can think of a transcription factor that can bind a small site, which is usually 6 to 8 base pairs or so, but its binding is so strong that it's stronger than a nucleosome, right? If it's able to bind and through DNA replication, so when the nucleosomes are removed, it grabs that DNA and competes with the histones. So the histones now can't come in and bind to this DNA, right? Does that make sense? So there was a nucleosome here. The replication removed this nucleosome, and then there was a pioneer that came and grabbed this. Okay? Now the nucleosome cannot form on top of this, right? But it's going to leave a bunch of naked DNA for the other transcription factors to come and bind, right? So this could be one mechanism. Give me another mechanism that we actually learned on how transcription factors regulate chromatin. We learned it in the eukaryotic transcription lecture. What could be another mechanism by which a pioneer factor can clear out nucleosomes from its binding site?
It can recruit other proteins?
Exactly. It can recruit chromatin remodeling proteins, right? One of the protein, the complexes it can recruit is it can recruit an acetyltransferase complex that's going to acetylate the nucleosome nearby. But this acetylation does not necessarily remove the nucleosomes, but we learned in the eukaryotic transcription lecture that the acetylation can serve as a binding site for ATP-dependent chromatin remodelers, such as switch-SNF, which can come in and literally take that nucleosome out, right? So these are ATP-dependent remodelers that were capable of removing nucleosomes. So these pioneer factors might indirectly or directly recruit these complexes to remove the nucleosomes. All right. And the evidence suggests that that actually indeed is the case for a lot of transcription factors. All right, so that's how nucleosome occupancy and positioning in the genome can regulate transcription by modulating transcription factor binding. And the second concept that we're going to learn now is that transcription factors often act in combination with each other to regulate the transcription of one gene. Give me one example in bacteria where different combinations of transcription factors modulated the expression of an operon so it could tune it up and down.
The
Lac operon. Lac repressor along with cap could regulate, right? So this type of combinatorial regulation of transcription could be independent, such as what we learned in the LAC and the CAP. That is, the transcription factors bind independently to their sequences, or they can have very close combinatorial action, which we're going to talk about in a minute. Okay? So why this combination is important? It's important because in humans there are about 25,000 genes. And so there are about 2,000 transcription factors. So every gene cannot just take one transcription factor for its regulation, right? So these transcription factors, first of all, are going to regulate multiple genes. But you also need combinatorial action because these genes need to be regulated differently in more than 200 or 10 cell types that's estimated in humans. And then these cell types are not static. They're going to have different transcriptional profiles during development, right? So the type of genes that we express when we were an embryo in our mother's bellies versus now as an adult are different. Give me one example of a gene that we express differently. Lactose intolerance. Right? We don't express the enzyme that are going to degrade the lactose, and some of you European descendant ones contain certain mutations to be able to actually still express that gene. I'm going to try and track down that paper, which is, I love that they actually could find what the mutation is that regulates that. But what I'm trying to say here is that even in a given cell type, you don't necessarily express the same genes all throughout development, all through different conditions, where like the summer versus winter is different, right? All right. So how does that then happen? So let's now imagine what the problem is here, that you need to increase the number of genes that can be regulated up and down with a limited number of transcription factors. So that's the problem of a genome, right? So what you can do is you can increase the range of regulation by tuning expression or using range of regulation, or also the targets of regulation by using different combinations. Let's say that there are 3 transcription factors. Here is just a very simple example from your book. And these transcription factors are A, B, and C, and each have their activation domain, and they have their specific DNA binding domain. And what you can do is you can use, if you require 2 transcription factors to regulate a gene, you can have 6 different types of sequences that these transcription factors can regulate, right? So instead of each going to one gene, you can have a combination. So what I mean by that is, for example, what you have is, in a given cell type, you want to express this gene, but not this gene, right? So if only transcription factor A was controlling this gene and this gene, they would be both expressed, right? But if you require that this gene now is controlled by 2 of transcription factor A, but this gene is controlled by the transcription factor A and B, and if that given cell type B is not expressed, even if the transcription factor A is present, this gene is not going to be expressed, right? So that's how you increase the range, increase the type of regulation that you can do. All right. So add into that certain other factors like an inhibitory factor, etc. Now you can have multiple different type of genes that might be regulated by the presence of different combinations of the transcription factor along with whatever inhibitory factor that might be, right? So that inhibitory factor could regulate the transcription factor binding, such as a ligand, or it could be another repressor, if it's present, that transcription factor cannot bind, or something like that. All right. All right, so just as the same example, for example, this gene here can be expressed only if both B and C were in the cell rather than only B or C. So this type of cooperation can occur closely or independently. So here's a close combinatorial function. I can't remember the name of these. Let me see if I see it. Yeah, so here we have two transcription factors called NFAT and AP1. So these transcription factors by themselves have very low affinity to DNA. Okay, so this guy can bind to DNA with low affinity. This guy can bind to DNA with low affinity. But when they interact physically, so this is a protein-protein interaction between these two transcription factors, it creates a certain conformational change at the DNA binding domain, and now these proteins can bind to DNA at a much higher affinity than they would by themselves if the other one was not present, right? So this is a very close cooperation where you actually affect the binding of the two proteins unless both of them are together. So here's another example of that, how that happens. There are also cases where the proteins, the transcription factors, do not physically interact, right? But their action on the enhancer or the promoter is cumulative, right? And such an example is the LAC repressors and the CAP activator. Their action was separable, right? The LAC repressor could bind without the CAP, etc. But they do, together their activity cumulatively affected the amount of transcription from the LAC operon. So in humans and in a lot of other eukaryotes, the enhancers are bound by multiple transcription factors, and the complex of the enhancer DNA with all of these proteins have been termed the enhancosome. Okay? What that means is that in humans, usually the individual enhancers are about 400 base pairs or so. You're going to have multiple transcription factors bind to these enhancers, and here are their sequence binding sites, and some of these binding are going to overlap, but some of them, the binding are going to be individual. And the activity, so how much transcription is this enhancer going to support, is going to depend the number and the kinds of transcription factors binding to that enhancer and forming that enhancosome. Okay? All right. So what are some concrete examples of this? The best concrete example of, in my opinion, how combinations of transcription factors can control transcription and regulate cell structure and function is in neurons. Some of you guys are neuro majors, so you might really appreciate this picture here showing you all kinds of different neurons of the pictures. And these neurons look like this because they express a different set of transcription factors inside them. And those transcription factors then regulate a different set of the transcriptome and regulate different genes at different levels. So how does this happen mechanistically, right? Here's one example how that might happen. So this is, these are two transcription factors shown here, LXH3 and NLI1. And so you have this transcription factor homodimerizing and also interacting with this other transcription factor, LHX3. And this interaction and the presence of these two transcription factors only are going to activate genes that contain the LHX3 binding sequences with a certain distance, right? And so presence of these two transcription factors are now going to be able to activate this type of neuron specification. I'm not a neuro person, so if I make a mistake, just jump in and correct me. All right, in another neuron, what you have is you have another transcription factor called ISL1 being expressed. Now, when this transcription factor is expressed, what it does is it now interacts strongly with NLI1 and also interacts with LHX3, and it also has a DNA binding domain. So when these three transcription factors are present, the genes that contain this kind of promoter sequences where you have two ISL1 binding sites that are at a certain distance and LHX3 binding sites that are now at a larger distance are going to be activated. So let's think of the opposite way. In the cells where only these two transcription factors are present, these genes are not going to be activated because the LHX3 binding sites are not located close enough. Right? Does that make sense? That you have, by having a single transcription factor that is specific to a neuron type, now you can target a whole different set of genes that have a different type of promoter structure. Does this make sense? All right, so here is a combinatorial action that specifies different types of neurons. All right. So transcription factors in eukaryotes can be regulated by themselves through the binding of ligands. And this could be through the changing of the DNA binding domain and getting into a conformation that is capable of binding, or it can also regulate the subnuclear localization of the transcription factor. For a transcription factor to bind and activate gene expression, where in the cell does it have to be? Nucleus, right? If it's sitting in the cytoplasm, it's going to do no good for anybody, right? So if you want to then maybe regulate a transcription factor's binding as a result of a certain signal, what you can do is you can sequester the transcription factor inside the cytoplasm until the signal comes in, and then you can send the transcription factor, right? And now we're going to learn about this in the context of hormone receptors. So these hormone receptors in humans is a big deal, right? Our biology and how we function really depends on hormones a lot. And some of the hormones are shown here. These are small molecules like cortisol, retinoic acid, and thyroxine that bind to certain proteins. These are called the hormone receptors. So the hormone receptor, are a family of proteins that act as transcription factors, and they have specific protein domains. So here is a general primary structure of a hormone receptor. They're made up of a DNA binding domain shown here, so that's found in all of the hormone receptors. Another domain that is called the ligand binding domain. So this is the protein domain that binds to the hormone, right? And then there's a variable region here that contains part of the transcription activation domain. All right, so examples of these hormone receptors are estrogen receptor, progesterone receptor, glucocorticoid receptor, thyroxine receptor, retinoic acid receptor, etc. So what kind of DNA sequences do they bind? They are shown here. So the DNA sequences that these hormone receptors, transcription factors bind are called the hormone receptor response elements. Okay, and here's an example for the glucocorticoid receptor. So this receptor binds as a homodimer. So one of the DNA binding domains coming from one of the protein is going to recognize this AGAAC sequence, and the other one is going to A sequence, and the other one is going to recognize the sequence on the other strand. And they need to be separated by 3 nucleotides. And when we want to say it can be any sequence, right? So it's denoted as N. Here it can be any sequence, but there needs to be only 3 of them, right? And the estrogen receptor binds to a different sequence, again, that is separated by 3 nucleotides. And you can see here these three hormone receptors bind exactly the same sequence, right? But their binding specificity is determined by what? Just look at here and tell me what is the binding specificity determined for.
The amount of nucleotides in between.
Yeah, the distance between the two sequences, right? So that determines the specificity of the hormone receptor binding. All right. So how do they actually activate transcription molecularly? So here is a crystal structure of the estrogen receptor activation domain that is bound to the estrogen, so that is the hormone, or tamoxifen. Tamoxifen is an antagonist. Okay, so the hormone is called the agonist. Agonist is a substance that fully activates the receptor that it attaches. An antagonist is a substance that attaches to the receptor but does not activate it or kicks out the agonist. Okay, so the tamoxifen, when tamoxifen is bound, this estrogen receptor cannot activate transcription. When estrogen is bound, it's able to activate transcription. Here, so the green alpha helix here is from the estrogen receptor that directly interacts with the ligand, that's the estrogen here. And that interaction basically stabilizes this helix over to the ligand, okay? And what that allows is that there is a surface here in the receptor activation domain that is interacting with the alpha helix from a coactivator. So transcription coactivators, give me an example of a transcription coactivator. Something that helps a transcription factor activate transcription. It's from eukaryotic transcription lecture. There's a huge one with so many proteins in it. There's a huge complex. Come on, one of you speak up. What are the proteins that help transcription initiation in eukaryotes? Mediator, thank you guys. Yes, a mediator could be one of the transcription coactivator. It could be a histone acetyltransferase complex, right? It could be an ATP-dependent remodeler. These are transcription coactivators. They help activate transcription. So what this estrogen receptor activation domain does is it recruits the transcription coactivator through this physical interaction. Okay? When the tamoxifen is bound here, what happens is that this alpha helix no longer is capable of interacting with tamoxifen. So tamoxifen structure is such that it does not allow this green helix to interact. And what this green helix does in its conformation is it actually prevents this alpha helix from the receptor to interact with the transcription coactivator. So when the protein is then now bound to tamoxifen, it's not going to have any interaction with the coactivator, so it's not going to be able to activate transcription. Does that make sense? All right. So that's how the ligand actually regulates the transcription factor activity. And there is another way in which the ligand regulates transcription factor activity, and it does so by changing the subnuclear localization of the receptor itself. Okay. So in hormone receptors, what you have is the receptor usually staying in the cytoplasm because it interacts with a set of proteins. Here they are labeled as inhibitors that inhibit their passage into the nucleus. So when these inhibitors protein are bound, and they're bound to the ligand binding domain, the protein cannot go into the nucleus, so the transcription is off, right? And when the ligand, in this case the hormone, is bound, these inhibitors are kicked out, and this protein bound to the ligand is now capable of being transported into the nucleus where it can activate transcription. And how do we know this? What kind of experiments do you do to know this? So again, you can use a reporter assay. Now we need to design our assay to test what part the question here is which part of the protein domain controls the subnuclear localization of glucocorticoid receptor, right? And you need to design a reporter assay to test this. And here I'm not going to show you all the different versions of the protein differences that people have done, but just show you the end result with a positive and a negative control. Okay? In this case, the negative control is just the reporter itself. So what you can do is you can express beta-galactosidase itself. As a positive control, what you can do is you can fuse the glucocorticoid receptor to the beta-galactosidase. When I say fuse, that means you're just making one protein that contains both of these. And then here, what you can do is you can take the domain of the glucocorticoid receptor and fuse it to the beta-galactosidase. And so this is the domain you're going to test. Is it controlling? Is that domain controlling the subnuclear localization? And now what you can do is you can express each of these in cells, and upon the hormone addition, you can look at the nuclear or cytoplasmic localization of the protein. And here's the example here. On the top, what we're looking at is we're looking at beta-galactosidase expression that is shown as white here. And what you can see is that in the absence of the hormone, in this case, the hormone is dexamethasone, so minus dex. In the absence of it, what you can see is the negative control, so the protein that only has the reporter, is mostly localizing to the cytoplasm because you can see lots of signal at the cytoplasm, but the nucleus is kind of dark, right? And when you have the receptor fused to the reporter, also the receptor is now only localizing to the cytoplasm, it's not localizing to the nucleus. Now, when you add the hormone, your negative control, nothing happens because there's nothing that controls the subnuclear localization of the reporter, and your reporter stays in the cytoplasm. But what you can see is that the glucocorticoid receptor, with the addition of the ligand, is now completely localizing in the nucleus, and you no longer see the cytoplasmic signal, right? So you know that this protein is sufficient in localizing here. Then you ask what domain is actually sufficient of doing that. And what you do is you do the same experiment with the reporter fused to the domain that you're analyzing. So this is the ligand binding domain. And you can see that ligand binding domain was sufficient in bringing that reporter into the nucleus upon the addition of the hormone. So you know that is the domain that is responsible for that subnuclear localization. So questions about this? All right. What other reporter could you use here that is visual, that you can see? What other reporter gene? GFP. And that's pretty typical. I mean, a lot of people use GFP for these type of assays now. All right. So these are the different mechanisms by which transcription factors are regulated. The next thing that we need to know in understanding how eukaryotic transcription is regulated is we need to know how enhancers and promoters work, because these are the gene regulatory elements that are actually going to control the transcription of a gene. So we went through this in the last lecture, but what we learned was that enhancers have so enhancers were these DNA fragments that can be anywhere near the gene or quite far apart, and there were different enhancers that can control the expression of the gene in different tissues or times, right? And we said, okay, for this gene, that where you have a reporter, you can look at which enhancer is controlling the expression of the gene in which tissue. For example, here's an enhancer that helps express the gene in the retina. So we said that you can find these by deletion assays. And when people did ChIP-seq experiments, now looking at chromatin modifications across the genome, right? And how can you do that? How can you use a ChIP-seq experiment to look at to map chromatin modifications across the genome. You could map nucleosomes with an antibody that's specific to the histone. What do you need to use to map a certain modification in the genome? What reagent do you need to generate? It's got to be an antibody, right? And the antibody should specifically bind to a histone if the histone has a specific modification. And these antibodies do exist, so you can make antibodies that's going to bind to a histone tail if it's only methylated in lysine 27, or you can make an antibody that's going to bind to a histone tail if it's only acetylated in lysine 16. Okay? So then what you can do is you can use those antibodies and map all the nucleosomes that contain that certain modification. Through these experiments, what the researchers have found is that enhancers have distinct chromatin structure marked by specific histone modifications. So now we can actually find enhancers quite readily in the genome by just mapping histone modifications. So the enhancers, their activity are regulated by transcription factors that bind to them, right? So if an enhancer is active, it can have transcription factors binding to it, and if these transcription factors happen to be activators, they're going to activate that transcription of that gene, or they're going to repress if they're repressors. And then presence of different transcription factors in the enhancer is going to determine how much the transcription of the gene is going to happen, right? But what we don't know so far, and still don't understand, is we don't understand how the specificity of enhancer-promoter interaction is initiated. That is, how does this enhancer interact with the promoter of the gene and control the transcription versus that enhancer interacting with promoter of another gene, right? Because we said that enhancers can be actually pretty far away, and in the human genome, more than a quarter of the genes have their enhancers that is a gene and a gene after. So you have an enhancer controlling a gene, but the enhancer is actually closer to another gene, right? So how the heck does that happen? Like, how does the enhancer know to make the right interaction, the looping interaction with the target gene? We still don't understand. But one thing that we know is we know something about the mechanics of it, right? So we learned about chromosomal looping, and there are some protein complexes that are involved in looping. Anybody remember these protein complex names? SMC protein complexes such as cohesin and condensin, right? So what we know so far is that the enhancers talk to promoters through DNA looping, and these loops are stabilized in certain cells and tissues by the cohesin and condensin complexes that are shown like this as a ring. Okay? So that's the mechanics of it is pretty well understood, but the specificity is not. But we know something about specificity, which is that there are these elements in the genome that act as insulators. That means kind of like barriers between the different regions of the chromosome. We learned about such insulator in terms of heterochromatin spreading, right? We learned that the heterochromatin in a chromosomal region, so let's say this is your chromosome and this is the heterochromatin here, the heterochromatin spreading can be blocked by the presence of an insulator so that the heterochromatin cannot spread and now repress this gene that happened to be on the other side of the insulator. So what this insulator is doing is it's making sure that the chromatin structure here does not affect the expression of that gene. So with the same type of idea, what we know is that these insulators also regulate which gene a certain enhancer can interact with. So if you have an enhancer that is in between two genes, so these are two promoters that it can interact, and if you have an insulator between that enhancer and this promoter, it actually prevents this enhancer's looping to this promoter so that now this enhancer can only regulate this promoter, right? So that's something that we know in terms of specificity, that the insulators can regulate which genes an enhancer is going to interact with or not. And so that's it for today, and I'll see you on Wednesday for more regulation.